Hello, my name is Sarah Robinson. I'm a third-year PhD student in the Oxford Protein Bioinformatics group, and I'm going to talk to you about my poster, which is based on our preprint currently on bioRxiv. You can see the link in the references at the bottom of the poster, entitled Epitope Profiling of Coronavirus Binding Antibodies using Computational Structural Modelling. In this paper, we've shown that by clustering the antibodies based on the similarity of their model structures, we can actually group antibodies which bind to the same site, the same epitope, with high accuracy. The motivation behind this research was that understanding the epitope of the antibody, the site to which the antibody binds to, is really crucial for understanding the function of the antibody. Epitope profiling, grouping antibodies which bind to the same epitope, can be done in laboratory assays, but most of the existing bioinformatics methods typically use sequence-based clustering, like clonotyping, where antibodies with high sequence identity are grouped together, assuming that then they are likely to bind to the same site because of this high sequence identity. And we know that antibodies which bind to the same epitope must have some similarity. They must interact similarly or have some structural complementarity to enable a binding to the epitope. However, many antibodies that engage the same epitope have dissimilar sequences, but might have similar 3D structures. So we've developed this new method based on the structural modeling of the antibody sequences and then clustering based on these structures to group disease-associated antibodies to the same epitope, regardless of how similar the antibody sequences are. Our approach utilized COVABDAB, the Coronavirus Antibody Database, which is a database of antibodies that bind to coronaviruses focused on SARS-CoV-2, but also containing SARS-CoV-1 and MERS binders that was created by Matthew Raybould in our group. In this database is sequence and structural data on antibodies which have been published or available in patents, and which we often have an epitope binding region for example, the spike, the S2 domain, N-terminal domain, in the case of SARS-CoV-2, if the data was included in the table. Now, not many entries in COVABDAB have solved structures available, only around 5%. Therefore, using the over 2,000 antibody sequences in COVABDAB, we built 1,500 high-quality homology models using a bodybuilder, and then we clustered these models. Our clustering approach first grouped antibodies with identical length CDR regions, and then we performed greedy clustering using pairwise structural RMSD values between the modeled CDR regions with a clustering threshold of 0.75 angstroms. The results from the clustering of the 1,500 models is that we get a set of 1,159 predicted structural clusters, where each of these is adopted by at least one antibody sequence from COVABDAB. 200 of these clusters were multiple occupancy, meaning the structures were adopted by more than one antibody sequence, corresponding to 541 antibodies that are contained within these shared clusters. And excitingly, 92% of these clusters containing multiple antibodies all bound to the same coronavirus domain. So this suggests if we have an antibody sequence and we don't know, for example, where on SARS-CoV-2 it binds, we can cluster it with antibodies where we do have the binding data and then predict which epitope this antibody likely binds to. In addition, nearly half of the multiple occupancy clusters contained at least one pair of antibodies from different clonotypes, which, as you can see in an illustration in figure 1, where here we've got distinct structure 19 containing 10 antibodies across 4 lenient clonotypes. So here our lenient clonotype definition is if the sequence has within 80% CDRH3 identity and aligning closest to the same V gene. We can also group antibodies from different species. So most of COVABDAB is human, but we also have mouse antibodies, human mouse, transgenic mouse, etc. And these can be clustered into the same structural cluster if they form the similar structures. In conclusion, our structure-based approach identifies functionally similar coronavirus binding antibodies that are too sequence dissimilar to be grouped by clonotyping. And by using the predicted structures of these antibodies, we can map them to the same epitope, regardless of the clonal origin or species that the antibody comes from. We think our methods provide an orthogonal tool to clonotyping to be able to functionally characterize the antibodies prior to doing in vitro assessment. We also think this shows that antibody immune responses might be more convergent than are suggested by the sequence-based approaches, as we do see these shared structures being adopted by multiple sequences. Thank you very much for visiting my poster. Feel free to get in touch with me if you have any questions, and check out our preprint that this poster is available on. I'd also just like to quickly thank the funders for this research and everyone in the Oxford Protein Informatics group.